What the scientists proposed to do would have seemed impossible 10 years ago. Of the thousands of genes in each cell of the laboratory rat, they wanted to isolate the single gene that makes insulin in the rat's pancreas gland. Then they would transplant this gene into bacteria, hoping to create a microbe with the insulin-producing ability of a mammal. If they could make it work for the insulin gene, it might become possible to make dozens of important hormones and drugs. A group of scientists at the university began to collaborate on the experiments.
So we obtained the insulin-producing cells from several hundred rats and extracted from them the chemicals from which we could get the DNA. Axel Ulrich took this material, inserted it into a plasmid, and placed the plasmid into a strain of bacteria that could only grow in the laboratory. These steps of the experiment were carried out under strict conditions of containment.
Howard Goodman is professor of biochemistry at the University of California, San Francisco.
Various methods. We had been working on the cloning of rat insulin for about a year. We had many technical difficulties and we began to overcome them. But it wasn't until the moment I saw this film, which was the analysis of the gene that we had cloned, that we really knew we had the rat insulin gene in the bacterium.
The next step is to show that the genes which we have transplanted to the bacterium can actually work and produce insulin. We'll then try to insert human genes into bacteria instead of the rat genes. If we can get these genes to work, this would provide a virtually inexhaustible supply of human insulin, much better than animal insulin since it would have no undesirable side effects. Eventually, in a generation or so, we might be able to do something which is really fantastic. It might be possible to rejuvenate the defective cells in the diabetics so that they would produce insulin in a normal fashion. Thus, the disease would be cured.
In the future, bacteria modified with recombinant DNA may be grown in industrial amounts. The bacteria could produce hormones like insulin and other important drugs. New fast-growing strains of bacteria or algae could be used in vast amounts as animal feed or fuel. A whole new technology will be needed to pick the desired bacteria. Automation will be used to inoculate, incubate, and photograph bacteria. The system produces thousands of photographs under computer control. These could be used to select the most productive bacteria for industrial use. Breakthroughs in genetic engineering may open up a whole new realm of biology. Until recently, it has been very difficult to identify specific genes on animal chromosomes. But with new techniques of gene splicing, scientists have begun to map the uncharted terrain of the chromosome. Stanford University biologists have made a remarkable photograph of fruit fly chromosomes. While trying to locate a certain gene on one chromosome, they found to their surprise that the gene's DNA code was repeated dozens of times. To explain this important finding, the scientists came up with a new idea. Just as each letter of the alphabet is found in many different words, each gene copy may play a different role in the life process of the fly. Biologists are now busy testing this idea. It could be the key to the miracle of growth, helping explain how a human being develops from a fertilized egg. The new molecular biology is leading to cures for genetic diseases. It may enable us to engineer our own genes and influence our destiny. It might even lead to complete knowledge of the secret of life. But is it possible to explain life in terms of chemicals and molecules? Or are there profound mysteries beyond the gene? While raising hopes of great advances in medicine and biology, recombinant DNA research has also created a major controversy. Tinkering with the stuff of life is not without its risks. Some people are afraid that a bit of DNA spliced into a harmless bacterium could create a deadly germ. Others fear that mixing genes from different species could have unpredictable effects on evolution. Government agencies have enacted strict controls for recombinant DNA research. Most scientists say that if these guidelines are followed, genetic engineering is not dangerous. With today's technology, we can only add a few foreign genes to bacteria. But 100 years from now, we may be able to mold genetic structure as freely as a sculptor shapes clay. The question is, will we be ready for the power to change life?